Okay, so the first part of photosynthesis is the light-dependent reaction. So the first step is the photoactivation of photosystem 2. So the photosystem 2 contains two special chlorophyll molecules, and then the light comes in and the chlorophyll absorbs the light, the photons of the light, and the electrons in the chlorophyll are excited, and two electrons are donated for each of them to reduce plastoquinone. And so there's a total of two plastoquinone reduced and 4 electrons lost. So after this occurs, photolysis is the next step, and this is when the chlorophyll from photosystem 2 is used to oxidize H2O, which is one of the reactants of photosynthesis. So this results in 2 electrons being produced, and these are used to replenish the electrons that were used in photosystem 2. And then it also produces 2 hydrogen protons, and additionally 1/2 O2 is released as waste. So the next step is the electron transport chain. So plastoquinone transfers electrons to a series of electron carriers, and energy is released as the electrons are passed, and the energy is used to pump the hydrogen protons across the thylakoid space into the lumen. And so because of this, the concentration of hydrogen protons builds up in the lumen. So the next step is called chemiosmosis, and this is when protons are moved from the lumen into the stroma. So the protons are moving from a high concentration of protons, hydrogen protons, to a low concentration. So because of this, it is called photophosphorylation, and the protons are traveling through ATP synthase. So this results in ATP being generated, and at the end of the chain, electrons are de-energized and transformed into plastocyanin, or PC, which is a hydrophilic electron acceptor. So then the next step is when photosystem 1 is photoactivated, and this happens in the same way that photosystem 2 is activated, where the chlorophyll molecules absorb photons of light. And then after this happens, the electrons from photosystem 1 are received by ferredoxin, which is the next acceptor. And after this occurs, the NADPH is formed. So this happens when the 2 molecules of reduced ferredoxin are used, and NAD plus is reduced when it goes through NADP plus reductase. So the final products of the light-dependent reaction are the ATP and the NADPH and the O2 that is released as waste. So the last part of the light-dependent reaction is cyclic photophosphorylation. So if the supply of NADP plus runs out or more ATP is needed, then the excited electrons in ferredoxin will return to the electron transport chain, and instead of being passed to NADP+. As electrons flow along the electron transport chain, more protons are pumped and more ATP is produced.
The light-dependent reaction. The next phase of photosynthesis is the light-independent reaction, and this begins with carbon fixation. And carbon fixation occurs in the stroma when ribulose bisphosphate, 5-carbon, is fixed to the enzyme Rubisco, and a carbon from CO2 is added. This results in the production of 2 molecules of glycerate 3-phosphate, 3C. Ribulose bisphosphate, or RUBP, is a molecule that is essential for carbon fixation, as its fixation to Rubisco generates the glycerate 3-phosphate using reduction. Rubisco is then the enzyme found in the stroma that binds to ribulose bisphosphate in order to produce glycerate 3-phosphate. Glycerate 3-phosphate is necessary for the reduction phase of the light-independent reaction. It's produced as a result of the fixation of ribulose bisphosphate and Rubisco. So after carbon fixation, Uh, the next part of the light-independent reaction is reduction, and this is when glycerate 3-phosphate is created by the addition of carbon and oxygen. The 2-to-1 ratio is reduced, and therefore hydrogen has to be added. So in order to do this, ATP and NADPH has to be used, and these were both products of the light-dependent reaction. ATP provides the energy needed to perform the reduction, and NADPH provides a hydrogen atom. So next is the fate of triose, and this is the number of turns through the cycle and regeneration. So 2 triose phosphates can undergo a series of reactions to produce glucose molecules in the form of starch and other organic compounds. If all triose phosphates are used to make glucose, then the supplies of ribulose bisphosphate would be used up. The majority of the triose phosphate has to be used to regenerate ribulose bisphosphate. In order for the Calvin cycle to continue indefinitely, ribulose bisphosphate must be produced as much as it is used up. So in order to reproduce RUBP, energy is needed. So an example would be if 3 ribulose bisphosphates are consumed to make 6 triose phosphates, one of those trioses would be used to create glucose or other organic compounds, and the other 5 trioses would be used to regenerate ribulose bisphosphate. So what happens to the amount of ribulose bisphosphate, glycerate 3-phosphate, and trioses after the lights are turned off? So because ribulose bisphosphate is produced in the stroma and is light dependent, the ribulose bisphosphate will continue to increase in terms of concentration. Therefore, fixation could continue to occur, thus continuing to create glycerate 3-phosphate. So while glycerate 3-phosphate and ribulose bisphosphate are still able to be increased in their, be produced, the trioses cannot. And this is because energy is required to create the trioses, and that energy comes from the light-dependent reaction in the form of ATP and NADPH. So if the lights are turned off, then the light-dependent reaction can no longer run, and ATP and NADPH are no longer generated. Therefore, triose would stop being produced when the lights are turned off, but ribulose bisphosphate and glycerate 3-phosphate would still be produced.